Thank you. And Michael, I have to say, I love the papers you chose for the reason that these were clinicians who made observations and really changed practice. This wasn't big data. This wasn't fancy meta-analysis and statistics. It was just pure clinical observation. So there's still a role for that. Also, I think this is true. I'm not sure, but I've been told that Jim Todd has a framed copy of a paper on his wall, which is the rejection letter from the New England Journal. So don't give up. If you don't get it published the first time you send it in, just keep going. So you'll see too that there's a theme developing here, which is a bit of a personal connection with many of these papers. So in 1964, doctors Epstein and Barr and their technician Bert Achong found a herpes virus-like particle in cultured cells from Burkitt's lymphoma. At the same time, in the United States, at Children's Hospital of Philadelphia, there were two investigators, Gertrude and Werner Henle, who were very interested in viruses and oncogenesis. And Adam Finn might remember that the Henleys were still around when we were fellows at CHOP. They had their lab on the 8th floor. Once in a while, you would run into Werner and speak with him. So Epstein shipped some of these EB cells to the Henle lab in 1964 to study them further. And the Henleys were able to demonstrate that this, in fact, probably was a virus, because when they took a cell-free extract from the supernatant of where these cells were growing, this supernatant was able to interfere with other viral infection in vitro, and that was demonstration that the cells were producing interferon. They were also able to show that this virus did not infect other cell lines that were easily infected by herpes viruses. In addition to that, they found that serum from Burkitt's lymphoma patients had antibodies to the antigens that were being expressed in the EB cells. And this was the very first link between a human cancer and a human virus. They also found that nearly all African children had antibodies to this virus, and that many children in other countries also had antibody, and virtually all adults in all countries had antibodies. But at the time, this was, in a sense, a virus in search of a disease, because nobody knew what the primary infection syndrome was, if there was one, and it's possible that there was not. So the Henleys were German Jews who left Germany in the 1930s, came to the United States. And after World War II, they had a mission to bring scientists from Germany to the U.S. to train. And two of those scientists that came over, one was Harold Zurhausen, who ultimately went on to win the Nobel Prize for finding HPV causes cervical cancer, and Volker Diehl, who became well known for work in Hodgkin's disease. The writing isn't showing, but that's Zurhausen on your left and Diehl on the right. The other person you need to see in this picture is Elaine. Oh, now I'm blanking on her last name because the writing isn't there. But this was a technician who worked in the lab, and on August 10th, 1967, she called in sick. And Volker Diehl spoke with her and said, you know what, Bridget, they called her Bridget, not Gertrude, but Bridget Henle was going to get pretty mad if you don't come into work, even if you're sick. So just come in. Let me take a look at you. We'll let Bridget know that you are a good, hard worker, and we'll send you home. So when she arrived, she looked sick. She had a rash. She had adenopathy, and he immediately diagnosed her with infectious mononucleosis. It just so happens that they had serum on Elaine Hutkin, that was her last name, in the lab, because she was one of the few technicians there that was seronegative to EBV. And in addition to that, her lymphocytes did not grow in culture. And you know, EBV transformed cells grow easily in culture. So Volker-Deal decided to harvest her lymphocytes that day when she was sick and she came into the lab and see what would happen with them. Now, I asked him, did you think, I wonder if her illness is caused by EBV, or were you just being a good scientist and collecting blood and deciding what to do with it later? And I really didn't get the straight answer. But after her illness, her lymphocytes now readily were transformed. They grew out in culture very quickly. And in this classic paper, which is The Forgotten Wisdom, you see here in the green, before she was sick, she was seronegative to EBV. And then after the illness onset in the blue, she was not only seropositive to EBV, but her cells also expressed EBV antigens. And then they had a relationship with investigators at Yale who had a serum bank on college kids who get infectious mononucleosis, and they were able to demonstrate that before mono, they were all seronegative, and after mono, they were all seropositive. And so the cause of infectious mononucleosis was discovered by serendipity. And that's where the three princes of Serendip come in. Serendip is an old name for Sri Lanka. This was a fable about these three princes who wandered around the island and kept finding things that they were not in search of. And that was then coined into the term serendipity by Walpole in 1754. So what does it take for serendipity to be a thing? It's three elements. One is agency, the second is surprise, and the third is value. And believe it or not, there's actually a typology of serendipity that's been offered. And I won't go into the origin of these names, but there's Mertonian, which is where a targeted inquiry solves a given problem by an unexpected route. So an example of that was Cade, who was an Australian psychiatrist who believed that mania was gout of the brain, and experimented with guinea pigs by injecting them with uric acid. But in order to solubilize the uric acid, he had to make the lithium salt. And as it turns out, it was the lithium in the injection that changed the behavior of the guinea pigs. I guess these were manic guinea pigs. I don't know. But anyway, there are other examples here. Röntgen, who discovers X-rays because he's playing around with Crookes tubes and finds a mysterious glow across the room on a photographic plate. But this is an example with the Henleys and Volker-Deal of targeted inquiry that solves a different problem. So they were looking for the cancer and the virus, and they found the cause of Epstein-Barr virus. I want to finish with one other example of serendipity. This is Mel and Gail Bosma. These were scientists at the Institute for Cancer Research in Philadelphia. They were interested in the ontogeny of the immune system in inbred strains of mice. And they basically had these colony of mice that they bled for once a week, and their blood was assayed for the various immune globulin fractions in the laboratory by radioimmunoassay. Well, they found completely accidentally that there were 4 litter mates that had no detectable IgG1, IgG2A, or IgM. And they went back and rebred the parents of those pups and ultimately developed what is now known as the SCID mouse. So these mice have no B cells, they have no T cells, and they do not make antibody, but they do have NK and macrophages and neutrophils. And it was later discovered that their problem is a mutation in a protein kinase DNA activated catalytic subunit, which is a protein that is involved in double-stranded DNA repair. So these guys basically don't make antibody because they can't complete VDJ gene rearrangement. And we now know this is sort of a leaky form of SCIDs. But this was the first mouse model of immunodeficiency. These mice were bred and spread around the world. They've been instrumental in learning about human stem cells, and SCID human mice were made, where you put a human immune system into the mouse, and then you can infect them with HIV and other pathogens. And it's led to many insights into vaccination and cancer. This is the original paper reporting the SCID mouse. It was in Nature in 1983. And the reason I'm showing you this is because I want to point out one of the acknowledgments was a guy named Gary Marshall, who at the time was a college student working in the lab. This is not quite as flattering as Michael's early picture from when he was a resident. This is when you spent time with a hair blow dryer to try to look like David Cassidy. Anyway, I was doing a summer thing between years in college, and I was the guy at the bench doing the radio immunoassay, looking for these immune globulins, and found a bunch of specimens where there was no antibody. And I thought, surely I have screwed up. I didn't put the reagents in properly. Went back and bled the mice and did it again. And long story short, brought it to the Bosmas, and they were like, this is something. So here is my conclusion. Everyone talks about Pasteur's quote, the prepared mind, but I like this one from Isaac Asimov. The most exciting phrase to hear in science is not eureka, but that's funny. So thank you.